we need to go way back in our memories to our previous biology class and re-examine and review that skeletal muscle contraction process, right? So, skeletal muscle cells, in order to contract, they needed to have an action potential occur along their cell membrane, right? So don't forget, the cell membrane has a transmembrane potential across it, meaning there's a voltage difference between the inside and the outside of the cell, right? So for skeletal muscle cells, a neuron would release neurotransmitter, specifically acetylcholine, which would bind to receptors on the skeletal muscle cell membrane. And the receptor was also a chemically gated ion channel. So when the receptor would bind to the chemically gated ion channel, the channel would open and ions would flow through the channel. Mostly we were looking at sodium moving into the skeletal muscle cell, right? So the addition of those positive sodium ions caused depolarization of the skeletal muscle cell membrane. And the depolarization was important because it was a change in voltage that could activate our first voltage-gated ion channel on the cell membrane. So voltage-gated ion channels open due to change in voltage, and the depolarization is the change in voltage that opens the voltage-gated ion channel. All right, so the important thing about voltage-gated ion channels is that they allow action potentials to travel because they're like dominoes. Once you open one voltage-gated ion channel, all of the voltage-gated ion channels on that cell membrane will open. Just like when you knock down your first domino, all the other dominoes will fall down. These voltage-gated ion channels are voltage-gated sodium channels. Right? So once they open, sodium continues to flow into the skeletal muscle cell. And eventually that traveling change in voltage went down the Ttubules, which were just places where the cell membrane of the skeletal muscle cell dived deep into the muscle itself. And eventually that action potential will hit the Ttubules, and right next door to the Ttubules inside of a skeletal muscle cell was a structure called the Sarcoplasmic Reticulum. The Sarcoplasmic Reticulum is basically a storage sac of calcium. Once that voltage zapped the T-tubule, it would also zap the nearby sarcoplasmic reticulum. And this caused the sarcoplasmic reticulum to release its store of calcium. So the whole point of that action potential was to get the calcium to be released into the cytoplasm of the muscle cell. The calcium was important because the calcium had to bind to part of the thin filament. So with muscle contraction, we have the thin filament and the thick filament. Right? During muscle contraction, the thick filament grabs onto the thin filament and pulls, and that's what produces the movement. So the thin filament was made of 3 proteins: troponin, tropomyosin, and actin. The calcium is needed to bind to troponin, which causes troponin to move. As troponin moves, it also moves the attached tropomyosin. When the tropomyosin moves, we now have binding sites that become revealed on the actin. So the actin is sort of like the largest protein in this thin filament, and it has these binding sites on it for the thick filament protein called myosin to bind onto. But the myosin cannot grab onto the actin and pull until those binding sites are revealed, and calcium was necessary for that process. So once the binding sites are revealed, the myosin can grab onto the actin and pull, and we get contraction.